You noticed in this talk that as we learned about these Nobel laureates, that they had to design new techniques, new methods, in order to get the new knowledge, to do what they wanted to do with it, to cure polio, eradicate it. Similarly, our next speaker, Professor Arnaud Comment, he's a distinguished researcher at the EPFL in Switzerland. He works in the new type of techniques labeled dynamic nuclear polarization. And essentially, with this techniques, you don't have to use drugs, you don't have to do surgery in order to find out about new metabolic disorders, know how to characterize cancer. So if we could welcome Professor Comment.
Well, thank you very much for this kind introduction, and I thank the organizers for inviting me here. I am very pleased to see all these young people still interested in science. That's good. So what I'm going to talk about is a technique that is quite, let's say, unique in the sense that we can probe metabolism in vivo in real time. And this is a pretty new technique. And I will just go step by step from the, let's say, the fundamentals of how to understand why we had to develop this technique to do this task that is probing metabolism in real time. Can I have the, thank you. And try to explain what we can do with this technique. So I'm going to try to stay on a level that is understandable to non-specialists in the field. So let's see if I can do that. So basically, what I want to start by saying, as you all know, that's very basics, is that as a living animal or human, if we're all animals, of course, but we need energy to perform vital functions, right? And like my son, like my son here, he's eating a piece of cheese as a good Swiss citizen, and this energy, to make this energy, we have two main tasks to do. That is basically breathing oxygen and eating and drinking. Okay, and once we eat and we breathe, we can break down molecules. That is called a catabolic process. And we can build new molecules. Basically, our body can build new molecules. It's called an anabolic process. And to do that kind of task, we count on ATP, which is the basic energy unit. You all know that. And as impressive as it is, your body produces the equivalent of your weight in ATP each day. So that is quite a factory, right? And to do this energy transfer, as I said, ATP is the unit that you talk about. So you start from, let's say, a molecule of ATP that is in your body. And what will happen is that this ATP can be used, for example, by ribosomes inside a cell. It's not very clear here, but it's a cell. And it's going to manufacture, for example, proteins, right? And to do that, it needs energy, and it finds the energy in this bond here that breaks ATP in ADP, so to create inorganic phosphate. So it basically cuts a part of the molecule, use the energy that is found in the bond, and produce proteins. That's the factory. And once this is done, this molecule of ADP and this phosphate, or inorganic phosphate, can diffuse inside the cell and will eventually find itself next to a mitochondria, which is a little organelle that will actually use fuel that you feeding your body with to actually rebuild ATP. All right, so that's gonna use fuel and make some waste in the meantime. And eventually, you will go back through the cycle and diffusion and go back to your ATP. So, well, again, if my son breathes, and eat some cheese, let's see what happened to get this energy. All right, so we have a little zoom on the cheese, and let's take a piece of cheese and see what can be actually taken out from there. And I have two examples here. So it's basically fatty acid, in this case palmitate, so it's a long-chain fatty acid, and lactose. Okay, so it's a carbohydrate. Well, that you find in cheese. And what is your body going to do with this cheese is basically it's going to try to reduce it. So two examples of catabolism, two catabolic processes, is one is beta oxidation. So this is how you're going to break down this long-chain fatty acids into smaller units that the body can use as fuel. Okay? In the process of beta oxidation, you will create energy as well, but I won't go into this detail. Let's say that you're going to create smaller pieces to use as energy. Likewise, the lactose, using the lactase enzyme, it will cleave this molecule into simple sugars like glucose and galactose is eventually transformed into glucose. And this glucose molecule will then be transformed into pyruvates through a process called glycolysis. Again, this produces energy, but you end up with pyruvates. Okay, so that's the basic of catabolism. And if we focus, I'm going to focus now on how the mitochondria is using this fuel to actually recombine inorganic phosphate and ADP to go to ATP. And we go really in the essential process of energy in our body as animals, is that we use oxygen acid, carbohydrates, so sugars, lipids, and proteins, so these three, and we produce some waste, CO2, lactate, ammonia, and urea. And after the catabolic processes, we end up with small entities that can actually enter the mitochondria, okay, so that's the fuel, and you end up with acetyl-CoA, basically, that will actually enter this essential step, which is called the citric acid cycle, or Krebs cycle, or TCA cycle, to create, again, it's the same kind of process. You have NAD+, it's a molecule that you attach a proton to through this cycle. I mean, you load it with an electron, and then you can create this oxidative phosphorylation step that is going from ADP to ATP. All right, so, and in the meantime, you can also, the acetyl-CoA can also go into another molecule that's called acetylcarnitine, that's in the muscle and in the heart in particular. You can store this in this form. So that's the basic of energy metabolism. So now, it's all nice, but how can you actually probe this process in vivo? And this is where NMR becomes handy. And we're going to try to see if we can actually see the kinetics of this energy production in vivo. Well, what's nice about NMR is that it can give really information on the metabolism of a specific substrate, and I will show you how. And it can also be combined with the imaging technique, which is the MRI technique, to really provide spatial information on substrate and metabolites. So you can have what is going on and where it's happening. And this is the power of this kind of technique. And here I draw the three little molecules that we were talking about before. And so you see the butyrate, acetate, and pyruvate. They're basically made of carbon, protons, and oxygen in red. One of the good probe in NMR is carbon, all right? Well, the only problem is carbon, which might be used as an advantage. I'll tell you why, but it's carbon is at 99% carbon-12 with no spin, and so it's NMR inactive, so you won't see it, all right? Carbon-13, however, has a spin 1/2, which is the one we like in NMR. I won't tell you why, but just keep that in mind. And it has 1% natural abundance, but you can label it. So you can select the molecules on which you actually have a carbon-13. Sorry, the color is not that great, but It's a little spin, so it's a little magnetic moment that you can selectively put on an atom. And that's chemistry. The advantage of having a low natural abundance means that what you inject is what you will see. You will not have any background signal. You will know that this is actually coming, the signal is actually coming from the molecule that you selected. And then what's fantastic about this labeling technique is that you pick a specific position in the molecule, say glucose, say number 1 here, and you see that through the process of glycolysis, it will end up in position 3 of pyruvate, then possibly lactate position 3, or acetyl-CoA position 2, etc. 3 and 4 here on glucose will end up in CO2 and bicarbonate, and all this information can be recorded. Why? Because NMR is a spectroscopic technique. What does it mean? It means that it has each carbon and molecule has a specific sign. It has a specific frequency that gives rise to a spectrum, okay? And this is due to the fact that, again, sorry, the color is not that great, but this is due to the fact that you have proton spins, and these proton spins influence the carbon-13. So if the molecule is slightly changing, it's part of its structure, then you will see a different frequency. So this is the signal for lactate, and this is the signal for pyruvate. And so you can really take advantage of that to distinguish what you're looking at. So, well, it has been done for a while to study energy metabolism in vivo. I mean, for a while, I mean, at least for a century. This is an example here in 2001. And it's this example I show, it's in humans. So basically, it's infusing labeled glucose, uniformly labeled glucose, in a human. And you see what you can see is a lot of things, glutamate, aspartate, and so on. And you can also follow the time course. You can see how each molecule is actually produced as a function of time. Well, I have nothing against this study. It's beautiful. Now, the limitation of that is that you only have access to really the concentrated metabolites. So you can only see really the big pools, so to say, when we have a really high concentration. And also the actual time resolution you see here, it's about 5 minutes, 10 minutes. So you can have a point 5 or 10 minutes when a lot of biological processes take place within seconds. So that was a bit the problem. And why is it the problem? Why don't we have the sensitivity? And I explain why here, is that NMR is proportional to the nuclear spin polarization. So on each spin, you have a certain magnetic moment, and this magnetic moment add up as one magnetic moment along the field. So you have to apply a magnetic field, which is represented here by this arrow, and it will slightly orient. But you have to fight, the spins will slightly orient, but you have to fight the thermal energy, okay? And to fight the thermal energy, you try to increase the field to get higher polarization. Nevertheless, as you can see here in this number, so at 300 Kelvin and 3 Tesla, okay, so in vivo, 3 Tesla is a clinical field nowadays, you have 0.00025% of polarization. That means you see very, very, very little amount of spins. If you increase your field by 9 Tesla, you have to put down $10 million, and you get a factor 3. Pretty disappointing, right? So now what you can do is you can lower the temperature. And then lower the temperature from 300 K to 1 Kelvin. You slow down the fluctuation. You tend to have a thermal energy that's much less, and you have a bigger magnetic moment, much bigger in a distant field, more distant field. And that's just at 1 Kelvin, 0.075%. Still, not that great. So what can you do further down is to say, well, I also have access to electron spins. So it's the same principle. magnetic moments, but those electron spins at the same field and temperature, they have a polarization that is 2,000 times more than carbon-13. Well, that's represented by the big arrow, which means that if you manage to go to 1 Kelvin, you actually have 100% polarization. That is very cool. And that means that you don't have to go, as for carbon-13, to try to go to millikelvin to actually get this very high polarization. You can see these two curves. This is a logarithmic scale in temperature here. So you go from 100 to 1K. And 1K, also it's low. It's easy for let's say, for physics to reach. You simply use liquid helium. Whereas here, it's very, very tricky to go down. All right, so then what do? You count on the interaction between the electron spin and the nuclear spins to try to force the electron spins to give their magnetization to the nuclear spins. And that you do, I won't explain exactly the mechanism, but there's an interaction via the dipolar field between the two. And you count on energy conservation. You give to the electron spins just not enough energy through a photon, meaning you give microwaves, so you irradiate with microwave your sample, so you give energy to your electron spins, but just not enough to allow him to flip, to change his polarization, so that he has to interact and steal the energy from the nuclear spins to make the flip. And that is what is called DNP, dynamic nuclear polarization. So far so good, but we're stuck at 1 Kelvin. That's not very biocompatible, as you can understand. And that is a toy that was developed by the physicists in 1960. So this is the technique that we want for biology, is to try to bring this sample now to in vivo, that is, to room temperature before you can inject it. And this is how it's done. This is a picture of our little machine that I built at the time. And I'll explain. I didn't invent it. I reproduced what was already invented, and I'll give the credit to the inventors just after I explain how it works, is that you take the substrate. So you take, for example, your glucose, and you add to this glucose solution. So you put glucose in water, and you add radicals. So you need electron spins. Usually electron spins do not exist as such in nature because they tend to pair themselves. An unpaired electron spin is called a radical. A radical is very reactive, so it usually finds a partner to react with. But you can find stable radicals, and you add a little bit, so you dope a bit your sample, you put it in the machine, you go down to 1 Kelvin. That's easy, as I said. You put liquid helium and you pump on it, and you irradiate with these microwaves to force the transfer. All right, so you do that for an hour or so. And this is the beauty of the experiment. The trick is, if you actually send hot water on your sample and you dissolve it very quickly, you actually can retain the polarization that you build at 1 Kelvin. So you trick the nuclear spins because they still think they're at 1 Kelvin even if they are at 300 Kelvin. And that, you can trick them for a little bit, and it allows you to increase your signal by 4 orders of magnitude, okay, if you compare at room temperature in the same kind of field. This was developed, as I said, by a company, Amersham Health, in Sweden in 2003, that was later bought by GE Healthcare. And I will tell you about the future of this technique just after, but just to go back to the limitation is that it goes back to equilibrium. So we created an out of equilibrium state by warming up the sample, and then it goes back to equilibrium with a time constant of about 1 minute. So in 1 minute, you have lost what you have built. So you got to be quick, all right? And first of all, you have to only select the nuclei that will have these properties. They're not all of them. So you've got to be quick. To be quick, we build this very specific setup. So we put the machine here, which we call the hyperpolarizer, and we do the protocol that I told you about. We send the microwaves and so on. Well, just the microwave for about 1 to 2 hours. Then we send the hot water on it, and we push very quickly with helium gas, with high pressure gas, next to, into an infusion pump that is next to the animal. That we do in 3 seconds. And after 3 seconds, we inject into the animal. Depending on the volume, it takes 5 to 9 seconds. And then we record the MRI. So I appreciate this is that we're going from 1 Kelvin to in vivo in 3 seconds. And that is the big trick to get high signal because it decays, and it decays rather quickly. So this is the technique. This is called hyperpolarization or dissolution DNP. And I'm going to show you now a couple of examples based on, I'm sorry, I guess it doesn't show, but we're going to use pyruvate and acetate and butyrate. So I'm going to show what you can see with that. So the most classical experiments, although you don't see it very well again, the colors are not great, is to inject pyruvate. So you're going to inject pyruvate. You're going to see pyruvate and lactate. And actually, this is in the brain of a mouse. You actually see every second how this transformation is taking place. All right, let me go to the next slide because this one doesn't come out very great on this projector. But here is another example. So we give another molecule that we talked about, acetate, to a muscle, a leg muscle, and we can see, so this is the acetate, so this is the signal taken every 3 seconds, I believe. And you can see that it decays with the T1, so it goes back to equilibrium. But in the meantime, there is another molecule that shows up, and this is acetylcarnitine. Acetylcarnitine is the buffer for the energy. So you can see in real time the enzymatic activity and how fast it is transformed from one molecule to another. Even better, if you do a little bit of modeling, and for those who have some biology background, they might appreciate that, is there is something that you call the Michaelis-Menten curve, is how fast an enzyme will transform an enzyme will transform a molecule from another as a function of the concentration. So by fitting these two curves, so the acetate and the acetylcarnitine, with a very simple model, but only 2 free parameters, we can actually deduce the enzymatic activity in vivo. So we can have access to how fast the enzyme goes. And this can also be done in the heart. This is an MRI image, just to show that we have our coil here. It's too bad because it doesn't really come out very nicely, the spectrum, but it doesn't matter. So basically, we do the same. We inject acetate into the heart, and you see actually the production of acetylcarnitine and also citrate. So it goes into the TCA cycle, and again, you can do that as a real-time measurement. And maybe like that, it sounds like it's pretty trivial. But you have to synchronize with respiration and blood pressure to get really narrow line. And what is really important here is that those rates of transformation are extremely sensitive to the disease state. For example, if you have an ischemic heart, meaning it lacks oxygen, you will see no acetylcarnitine whatsoever. So it will really be a dramatic change. And this is an exam that can be done in over a minute. And that is extremely important for the application. Here's another example. If you inject pyruvates in the heart, you can actually measure lactate, alanine, bicarbonate, and CO2 also, the production of CO2. Here is the production of, here is the CO2 signal as a function of time. Here it's 5 to 35 seconds. Here is bicarbonate. And you can deduce the pH. So you can measure, first of all, the pH variation as a function of time every 3 seconds. More importantly, you can really see what's going on in terms of how the energy is used. For example, if you compare a rat that has been fed normally with a rat that has been fasted, you don't see it very well here, but this bar here is the bicarbonate. So same rat, It's no disease. You stop the food for about 8 hours, and then boom, no more bicarbonate. So quickly, the rat with the heart of the rat will go from using the glucose that he has to switch to metabolism, to fatty acid metabolism. So you can really probe how fast the heart will react and use the energy. And we can also do that by injecting butyrate, so a fatty acid, and we again see, in that case, quite a few things like acetylcarnitine, acetoacetate citrate, glutamate, and so on. So you have access to all these metabolites and how they're created in real time. Well, there are many other applications, but one of the most, well, let's say, important so far in the field is for cancer detection and treatment response. Because as you may have heard from the previous speakers, in cancer, in most cancer cells, there is the so-called Warburg effect, which means that the cancer cells tend to eat a lot more sugar than others, and they tend to produce a lot of lactate. So you have a high lactate content, and by actually infusing hyperpolarized pyruvates, you can actually have a ratio between pyruvates and lactate in a specific area, so you actually can map that in specific area, so you have the spatial and the spectral information, and you see that if you check afterwards, so you see the map actually of the lactate image. So you see here, in this case, it's a cancer that was grown in a rat, I believe, and you can see the lactate map is really bright. So you can see the high lactate, and here you can see after histology that what has happened, you had very high lactate signal in a late-stage tumor, and even in an early stage, you already have a lot of lactate. So you have access to the lactate as a function of the disease progression, and that is a key thing. And finally, I wanted to mention this, is that it's a technology that's on its way to clinical applications. We were commissioned by the NCI NIH in the US to write a paper, a white paper that's called Analysis of Cancer Metabolism by Imaging Hyperpolarized Nuclei. And it was a goal to have a consensus between academia and the industry, because you need a lot of money to develop this technology for clinical applications, as we heard before, for most in vivo for human applications. And there is currently the first human trial at UCSF in the US. So the conclusion is that what I wanted to present here is that it's a pretty high-tech methodology here that allows this unique kind of biomedical imaging to probe in vivo real-time metabolic imaging. And this dissolution DNP, which is this technique, is on its way to clinical application. And maybe one of the thing that we can learn from that is that really this is the kind of science that requires many, many skills, more than most actually, because really you need the technology from the physicist. You need the engineering to build the best machine you can have and to go as fast as possible, because you have a temporary state of hyperpolarization. You need a chemist to make all the molecules, to label and to make the radicals, the biologist to tell you what is going on, and certainly the medical doctors to apply this to humans. Okay, so I just wanted to acknowledge all the people who work with me to get this data and to work on this project. So Rolf Grütter, who is director of the Center of Biomedical Imaging in Geneva, Lausanne area. The three postdocs, Jacques van der Klink, who started the project with me, the three postdocs, Mor Mishkovsky, Yuhei Takado, and Najat Salameh, and the 6 PhD students who are working here, Tian, Jessica, Ricardo, Tim, and Andrea, and Emine. And I would like to thank the people who give us the money generously. Thank you very much.